Can everybody hear me okay? Yep, great. Well, welcome. Excuse me. Thank you for being here today. Oh, excuse me. Sorry, I don't have COVID. So it's great to talk with you about a little bit what we've been doing about measuring and how to improve how we measure impacts of invasive species. So to give you a little bit of background, I want to start with this figure that is something that was put together by an intergovernmental panel from the United Nations, the IPBES, that had more than 100 scientists working on invasive species. And they defined impacts as these intersection of the impact on nature, which they define as everything that does not include humans, and then the impact on the quality of life of humans, as well as on the contributions of ecosystems to humans. So there's like these multidimensional aspect to impacts. Today, we're going to be focusing mostly on what we think about nature. So these will be mostly ecological impacts of invasive species, and not as much about the other aspects, but it's important to consider them as we move forward. So when they got together and put these large report, they look at more than 700 papers from across the globe, and not surprisingly found that invasive species are widespread. They are in every single ecosystem. They are in terrestrial and freshwater, as well as marine. Some areas of the globe have more alien, as they defined alien, not invasive or non-native. For example, North America has a really high number. They also found that locations like islands usually would have higher impacts. When they quantified those impacts, they determined that those impacts were quite significant, that in terms of cost, the cost of managing and the impacts of invasive species had more than quadrupled over the last decade. Now, this value might be a value that has a lot of variation around it, and how that value is estimated, it can be a topic of discussion for many, many hours. Nevertheless, what we can see is that it definitely is going up. So we definitely are investing more and more money in managing these invasive species, and we are concerned about it. Also, at least 80% of the impacts were considered to affect the quality of life, of human life, and with some of them having a positive impact. So there's significant impacts to humans, or at least we think so. So that means that our jobs managing invasive species are really worth it, and there was a call of action from this. So what happens with actually the impacts across species? So they found that invasive species were associated with local extinctions, at least 1,200 of those local extinctions, and that those local extinctions were caused by a variety of taxa, mostly vertebrates. So those are mostly caused by rats and cats that are introduced into islands and are associated with extinction of, for example, birds and native birds and things of that sort. But you can see that there's very few that have been caused by microbes. And the question then is that because we have not studied them as much, or we cannot identify them, or because the microbes are not causing those extinctions? And then plants are around 15%. So that's a significant number. And one of the questions we had then is like, how reliable is this data? Can we really make conclusions? So we asked ourselves, we're going to jump this one. How strong are the data supporting these conclusions? Have we really have a strong data backing up these big conclusions that invasive species are leading to extinctions and significant negative impacts across the globe? And obviously we could not go ahead and review every single paper that they did across the whole globe and every single type of invasive species there is. So we decided to focus on what I know best, which is invasive plants. And we took 3 plants, garlic mustard, tree of heaven, and Japanese knotweed. You can see the pictures in right there, like probably something that's very familiar to all of you. These are very widespread, non-native plants across the northeastern, and also plants that have been significantly studied across the globe where they are invasive. So you can get a lot of papers and literature that has attempted to quantify their impacts. So what I did was I took these plants and did searches for papers that measure the impacts of each one of these species. I use multiple databases, and from these databases, I extract every single paper that attempted to measure an impact from one of these species. Once I had that paper, we went through each one of those papers and extracted some information. So overall, we reviewed 235 publications, which amounted to 1,400 studies. So 1,400 variables that people had measured to assess the impact of invasive species. Once I had the paper, what I did was get some publication information, who did it, where, was it in a forest, was it in the lab, was it in a greenhouse? Then I collected some information about the characteristics of the invasion. And these are things regarding how long has the plant been there, how spread is it, how abundant is it. I have to say that information in most cases was not present, right? Either because the researchers didn't know, because we don't know when the plant first arrived, or it was not reported. Then I looked at how they did the study. Was it observational or was it experimental? Did they manipulate variables or did they went and measure in the field? And then what did they measure? So those are those response variables that I'm going to call metrics. So those response variables are what did they measure and how did they do it? And we're going to go over that in a second. And then whether they just measure invasive species or they actually put it in context of other things that could be occurring in the environment. For example, are other invasive species present? Is there deer herbivory? Are there pollution? Is there pollution? How are these affecting with climate change? So we were looking at these correlation with other variables around. So once I had those, I summarized them. And here are a few of our preliminary results. So on the, do we have a pointer? No pointer, so I'm just going to point.
There are some sticks there, I think.
That might be a little bit too long. So anyway, on the x-axis here, you just have the 3 species that we're talking about. So tree of heaven, garlic mustard, and Japanese knotweed. And then the first panel is the type of design that people use. So people either use experimental or observational, and you can see that it's pretty similar. And then you can see on the second one that most people use field experiment or field studies. And most field studies tend to be observational, and they relied on comparisons of invaded versus uninvaded habitats. As the ones on the picture, you have a lot of swallowwort on the top and no swallowwort on the bottom. When people relied on those comparisons, more than often, very commonly, they did not establish how far away these locations were or what were the characteristics of these different locations. So we really don't know how well these locations are controls of each other in many, many occasions. And finally, the last one, and we're going to talk about this one a little bit more in a second, is about the amount of time the study took. And you can see that the majority of the studies were done in a snapshot. So researchers went once, took one measurement, and from there concluded. And if you work in the field, you know that seasonality, year-to-year variation is quite important, as well as how the impacts accumulate or deaccumulate over time. So there's very, very few studies that actually had taken more than 5 years or actually gone for multiple times. So that is like a big overview. So what are people measuring? So people are taking measurements at 3 organizational levels: the population level, so that's at the species level, single species; the community, so that's a multiple species within a location; and then ecosystem functions, usually related to functions of the soil or the water. So what are people mostly measuring? In these graphs, what you are seeing are the number of studies where people measure each one of these variables. The darker blues are, we measure more of those, whereas the white will signify that we did not measure those. The first row, the first column are the 3 species. So we have the tree of heaven, the garlic mustard, and the Japanese knotweed. So at the population level, so when they were measuring impacts of a single species, they mostly thought about performance. What do we mean by performance? The size of the plants, the size of the leaves, the biomass that the plant produced, how tall the plant was, were usually what people measure the most. Then the second most common one was germination. And one aspect that was really important about germination is that most of them were done in the lab in petri dishes. So there was one seed placed in a petri dish with a struct of the plant, with or without the struct of the plant, and then look at the germination. But field studies of germination and how those are affected in the plant itself are going to be harder to find and less common. Now, we also looked at community measurements were mostly focused on abundance, richness, and composition and diversity. So these are very known measurements of diversity, plant diversity, or diversity around a location. Importantly, when we think about composition, so that's community composition, a lot of the studies found a difference between the composition of plants in a location that's invaded versus not invaded, but very few attempted to explain why do we see those differences in plant communities. So if the communities are different, why are they different and what's the mechanism that's making them different in any way? Finally, with the ecosystems, it was mostly related to soil minerals, and they were mostly assessed as mediating factors. So are these sites that have lower pH, have higher impacts, for example, or the impact of the plant lower or affect the amount of nutrients that are on the soil? Without that connection to whether the plant is actually using those nutrients that are on the soil or not. So overall here you see that there's things that we tend to measure a lot and things that we tend not to measure. There's very few, for example, demographic measurements. Demographic measurements are going to tell us how the population is doing, whether the population is increasing or decreasing. And we have very few of those when that should be something really important, because ultimately what we care about is whether invasive species have an effect declining the populations of native organisms. So we don't have a whole picture. We tend to measure the same things over and over again. And there are things that we measure in one type of venue, only the lab versus the field. And we fail to connect to other parts of the ecosystem. So then we went ahead and said, okay, if I go through each one of these papers that measure abundance of diversity, how many of them are going to support the assumption that the invasive species has a negative impact? So I went through the paper. The paper assumes, for example, that garlic mustard is going to produce negative impacts on, it's going to reduce the diversity of native plants. They went out and measured. Did they find that or did they not find that? So I went through each one of those papers that measure abundance or diversity and got that information. And then I went ahead and looked at that probability and asked, how is that probability? So a probability of 1 will indicate that we always found that matching, right? That prediction of negative impacts always happened. A probability of 0 will indicate that expectation of negative impacts never happened. So the first thing I want you to notice is that all our lines are overlapping with that probability of 50%, meaning that finding a negative impact of the invasive species was mostly a coin toss. Like you could find it or you could not, but we don't have overwhelming support for either of these 3 species indicating that their impacts are negative. Overall, For garlic mustard, these tend to be a little bit lower on the lower side, across an average, across 25%. So only 25% of the studies that quantified impact of garlic mustard actually found evidence that it was producing negative impacts. Some of them found no impacts, and some of them found even positive impacts. For Japanese knotweed, is the only one that where we found that some differences depending on the venue. And those were that in the laboratory, actually in the greenhouse, they found less impact. And I think that's because of the type of studies of Japanese knotweed that were done in the greenhouse. They were usually studies about allelopathic effects, but they were in larger pots, so those really did not manifest themselves, whereas they will manifest themselves in laboratory experiments where they're in petri dishes in very contained, very secluded spaces. So you can see that big assumption that we have that invasive species are producing negative impact, it's not really as supported by the data as we expect. So some of the common shortcomings that we observe is a very narrow focus. A lot of the species that we observe, like there's a focus on certain species. So we know a lot, for example, about invertebrates in Japanese knotweed, but we don't know about invertebrates in tree of heaven, for example. And we tend to have lots of information about microbial communities in garlic mustard, but not on the other species. So we still lack that like larger focus across taxonomic levels and hierarchical levels too. We have very narrow temporal and spatial scales. We talked about these already, but there's very few studies that span over decades or more than a year. And also the majority of these studies, a single study is obviously localized, but across the whole set of studies, we're still not covering the whole area of the spread of the invasive species. And there's a lack of scientific rigor. I talk a little bit about that, for example, having controls or references that are far away or not really similar to where the location is invaded. Ideally, we would like to have studies that are before and after the invasion or an invasion front. And the majority of studies are not using that type of design. Sometimes there's no true replication, or we have one answer inside A and one answer inside B. And the big conclusion is, yes, there are impacts, but it's quite site dependent, and that has not been communicated through the paper. And the last one is this lack of validation. A lot of the metrics that we're using could be dependent on many other things that are going into the system and are happening at that same location. So we are not establishing a clear link between the impact of the invasive species and what we're measuring. So at the end of the day, I'm still asking myself, is it that these species have no impact, or is it that we're not measuring it correctly? And how can we measure it better, in a better way that actually gives us an answer where we can untangle those? So moving forward, we want further taxonomic representation. We want measurements that are going to be easily measured and across the spatial and temporal scales and going to tell us similar stories when we measure at the local scale versus regional scale. We want measurements that allow us to understand what else is occurring in the space, because we know that invasive species are not occurring in a vacuum. There's multiple other impacts that are happening at the same time in the same location. So we want to make sure that we address those and that we understand how these mediating factors affect the impact of the invasive species, or if the invasive species is actually the driver of the change we see. If it's not the driver, then maybe from the management perspective, we can do something very different. And finally, I would really advocate for having metrics that are validated, that is tested and applied in multiple contexts and telling us the similar information across those multiple contexts. And that's what we think what we've been developing on the sentinel approach, which is what Bernd is going to give you a little bit more of an overview, and we're going to see a few examples of how we're applying. And that picture there on the right is an example of how I use the sentinel approach to look at the impacts of pale swallowwort. So overall, we think that we know a lot about invasive species, but impacts, but we don't. And we need to develop better metrics. And one of those approaches might be the sentinel approach. So with that, thank you all for coming, and I'll be happy to take any questions. Any questions?
I'm going to switch the slides, but go for it.
Analysis of the number of studies, did you take into consideration like the N value, like the how many replicates per study that they have?
Yes, so in the modeling approach, we included a random effect according to the study. So some studies, some publications had multiple studies, some publications had a single study. So we included that. So that's accounted for. Yeah. Yes.
So I saw that in your one figure, you were talking about the length of time, and that Atlantis had probably only a single more than 5-year study.
Mm-hmm.
Do you think that that's like a bigger issue than having fewer 5-year studies for something like garlic mustard, which is able to
Biennial? Yes and no. So on one hand, you have a tree that's going to take many years to accumulate, and the whole lifespan of that tree is definitely not going to be capturing a single snapshot. But also it depends on how long that plant has been in there, right? So if you're thinking about an invasion that's fairly new versus an invasion that has been happening over 10, 20 years, you might be thinking about different levels of impacts. So I don't think it's necessarily given the life stage of the plant, but more related to the length of the invasion and how big that invasion is, because the impacts accumulate over time. Or sometimes they accumulate. For example, in garlic mustard, we know that across time, when garlic mustard is present, it starts declining. The population becomes more and more sparse. So you might find very strong impacts at the beginning when the plant is really abundant, but then as the abundance of the plant decreases naturally without any intervention, those impacts might decrease. So that long term will be really equally important for a plant with all life stages, different like natural histories.
If you have other questions for any of the speakers, we will have a discussion time afterwards. We do want to keep moving those. I'm going to welcome Bernd Blossey to speak next.
Good morning, everybody. So slightly different title, same content. I'm happy to see the interest. Obviously, you all feel the need to somehow document what's going on and what you do and what the outcomes are. And so that has been a struggle for us for a long period of time. So I'm going to give you a little bit of an overview if I can manage this. So what we think is happening when we have an introduced species come in, it changes ecological relationships and it changes our relationship to monetary values that we have, right? I'm not going to put up any linkages in here because it's going to be just a spaghetti graph thrown at a wall, right? But we can see all kinds of linkages, and humans are somewhere in there, and how we interpret these changes it depends on who you are, what you're interested in, what your job is, and how you use introduced species, or how you are actually trying to combat that. So it's a network of relationship. And then once we start looking at that, we are confronted with our own ecological dogma that we have. And then we get ecological surprises. I'm going to give you a little tour of how we get to where we are. When we had the initial online metrics workshop, right? Some of you that participated threw all kinds of things out there, what we could measure, what we may want to measure. There is a short list of all kinds of things that may be coming out. The question then is, these effects that introduced species are supposed to have, are they common for all introduced species? We don't know. Or were they just documented for a subset of them, or maybe only one species? And the question is, what do we really know? And Andrea just gave you the answer. We may think we know a lot, but in the end, she used 3 species that are very well represented in the literature, where the answer is, not sure what the outcome is of that. We can't have generalizations. And for most other species, we actually know less than about the 3 species that Andrea looked at. Yeah, this is the dogma that we see, right? Introduced species, overwhelming threat. IPBES just came out with that. Then our assumptions as managers is that reducing abundance reduces impact. So that should benefit native species or monetary values, whatever. Yeah, that's not widely accepted. So we get into this dilemma of don't judge species on their origins, or if we do manage, we don't necessarily benefit native species. I'm not going to go into a lot of details, but you know that there is a debate out there, maybe not among the invasive species managers, but as soon as you get out of that little in-group, it's not necessarily widely accepted that there are negative impacts or that the negative impacts outshine the beneficial impacts. So if we summarize it, Andrea did some of that. Impacts are assumed, poorly documented. Perpetual control is necessary at high expense. You all face that. Do the desirable species return? We don't know. And then the impact of management is rarely assessed. Some outcome seems to be that threatened and endangered plant species appear to do better in the presence of invaders than in areas with herbicide use. So for loosestrife, one of the first examples that I worked in, the state of Minnesota stopped spraying because the native plant species were more negatively affected by the herbicide than by loosestrife, for example. So then we have inappropriate metrics to assess success. You've all seen that people use hour span bags of garlic mustard, others remove. Brian just gave a little talk about that. So it's a field critic for, or field day for the critics that argue we all just xenophobes and non-targets effects, that's overwhelming everything and it's waste. So that may not be the entire thing. So both I want to stress what Andrea said. We are not saying introduced species don't have an effect. That's not what we're saying. But what we're saying is the database that we are judging this on is really, really shaky quicksand, right? And so that's not a solid platform to then develop policy. And so we need to increase expenditure for that. And then when people do things like what Andrea did, do literature reviews, people come to very different opinions about what's actually out there. This is coming from a review of aquatic plants by Tasker, and don't have my reading glasses on, but just a couple of years old. Looked at some of your favorite introduced species, from hydrilla to phragmites to typha to milfoil. And the response is actually not what you would expect. Actually, some of the native species seem to be doing better in hydrilla, meaning on the right side of the dash graphs. Same for insects. So it's not as clear cut. This is based on what the literature shows, not how well the studies were done, right? That's always a thing. You can do all kinds of reviews and meta-analyses, but the outcome of what your review is showing is only as good as the data that went in. And that may be a problem. That's why we're here, for example. Then the assumption of beneficial effects here of management, this is herbicide, but people actually followed up. This is like 40 lakes in Wisconsin, and they looked at the communities that are, or the plant species within the communities that are favored, which are the ones that are in green, or the ones that are declining with herbicide use, that's the one in red. So for me, this graph is that, well, there's some hope for some species, but the vast majority of the species is negatively affected, not just milfoil. You're doing more than just milfoil control, right? So we need to be aware of these unwarranted effects that we have, or the revenge effects. Some people call it revenge effects of going and doing herbicide treatments or other management. Here's an example from Andrea's work where we needed to include, I think these sticks are too short, too long. I think they are good. I can reach out. So the important thing that Andrea did is including Jeremy Collins was the primary author. This is out in Ecosphere in 2023, so you can read that up. Worked with the forces in state parks, looked at swallowwort management, and included deer exclusion in the entire design. And what you can see here is that if these are the managed populations and here are the unmanaged ones, over time, swallowwort declines over the 3 or 4 years that they measured it. It declines more so when you fence the entire thing. Yes, with management, you can drive swallowwort down to very, very low levels. Not when you do not manage it, but with deer exclusion, you drive it even further down, right? The important thing is over here, the native community or the plant community response. Only if you do not manage do you actually get a decrease, a change in cover, or an increase in the diversity of native plants. So if you constantly interfere with doing something, the native plant species do not benefit from the activities that you are doing. If you don't do anything, they will respond, but you need to exclude the deer to actually get to that response. So this is what we like to see, right? Andrea talked, well, like to see, I don't know whether we like to see that, but this is what we assume is happening out there, right? The abundance of an introduced species changes whatever we are interested in, the diversity, survival, growth, or any other factor that Andrea before went. This is what we assume, but it is always a straight line, we don't know, but that's the assumption how we start out with. So when we started our excursions into these assumptions of impacts, we came up with a couple of things that were puzzling our mind. Andrea's first foray into garlic mustard showed that the ground beetle communities are really not negatively affected by garlic mustard. That paper was outright rejected. Boom. So I rarely argue with reviewers about things. And there I argued, why are you rejecting that? Because they're saying, well, you don't find the negative impacts of garlic mustard. So go critique the methods, go critique the analysis, not the outcome. So the editor said, all right, so it was published, right? So that was the first thing where garlic mustard should have had negative impacts. We didn't find them. Go and work with John Mertz, Vicky Nuzzo. Salamanders are not affected by garlic mustard, barberry, or Japanese stiltgrass. That's when we discovered introduced earthworms that are the driver of much change. Native plants is Vicky Nuzzo's work at Fermina, respond positively to deer removal, but are not affected by garlic mustard that came in, increased in abundance, and then declined. And then something else, aquatic invertebrates and tadpoles respond to species traits, particular carbon, nitrogen, phosphorus ratio, lignin, and others, but not origin. So there are puzzling things that we were finding by doing experiments, both in terrestrial and aquatic contexts, that then said, what is going on here? The idea of native species driving down the quality of our habitats or affecting native species doesn't seem to be materializing in some of the things that we do. But you can find these things out in the field. Brian Green's work in South Carolina. So very clearly showing that as privet cover, Chinese privet cover increases, the cover, number of stems per meter square, native species richness, or maximum height all goes down. That seems to actually support what we were saying. Hey, Chinese privet is reducing our native communities and decreases the health of them. However, important to recognize here is that the cover of privet is a function of urban development. So there is an associated thing that we have, and Chinese privet density is a function of the distance. Brian was the first to use the sentinel approach for a very different question. If you want to know more about his work, he's sitting right there. He can answer all those questions if he remembers all the details. I don't even know how we got into the sentinel approach, but we needed to try to understand what are the interacting factors that would drive the deterioration of the floodplain communities in South Carolina. So, okay. Or what we then know after 25, 30 years of loosestrife biocontrol is that the stem density of loosestrife, or that the total cover of native species is a function of the stem density of loosestrife. Now, this is not a straight line, and you can go down to 20 stems per meter square, and the cover of native species is still just lovely. You don't need to have eradication, right? But if you drive loosestrife down to lower abundance from something that was very high, that's per meter square, you benefit the native plant, community. So we think loosestrife was a driver, at least for plants. Then we have the ecological surprises, right? Deer exclusion over time reduces earthworm abundance. We still don't understand the mechanisms. That's not just our work. Other people have found this now in northern Wisconsin, in Ohio, and in many other places. So that's Elizabeth Simpson's work. So this is, when you put up a fence, you don't only change deer access, you change all kinds of other things and the effects that other things are having. And we need to be aware of that. We can't change that necessarily, but we need to be aware of it. Here are other things about microstegium. It's on that side, and garlic mustard. If you fence it, similar to what happens with swallowwort, the populations goes down. You don't need to do any other thing other than taken deer out of the equation. So remember that I said abundance or presence of an introduced species should drive down the performance or growth or whatever other metric we want to have of native species. Shauna Kay here did a little reductionist experiment looking at the diversity, which is not shown here, but also the development of American toad tadpoles in litter from different native and introduced species. So what you have here, the native taxa are in the gray and the non-native taxa are in the white bars. So what you would anticipate is that both the survival, the number or the proportion that metamorphs or the biomass index or so would be survival would be lower, metamorph production would be lower in introduced litter, right? You don't see any of that. So Jeremy Dietrich did the ultimate experiment for all of that. This is a little experimental design, 300 mesocosms, 125 liters, and then he put these wonderful little aquatic organisms in there. We had the lovely possibility to use both introduced and native species. So these are congeners, right? Taxonomic pairs, to not assume one or the other. We created a standard community of 7 species, created an invasion gradient from no presence to then higher and higher proportion of litter of introduced species until you had a monospecific community. We had these aquatic invertebrates and assessed the abundance over 2 field seasons. And this is the outcome. Again, if you think about the pairs, the introduced species should drive the abundance of whatever organism you have down. And the stronger your invasion level is, the more you would see that. None of that materializes, right? The 2 species are basically, for each phylogenetic pair, basically indistinguishable. And even if you go and have a higher invasion level, we don't see the decline in the abundance of native species. So what the hell is going on here for aquatic organisms, right? What is driving this? Because we do see change in these individual mesocosms that are differences in abundance. So, well, it's plant traits. All of these organisms respond in very similar ways to the CN ratio, phosphorus-nitrogen ratio, total nitrogen or total phosphorus, right? So it depends a little bit, except here for the CN ratio, these are the different species, what they're doing. And these are the responses that the organisms are having. They're all responding very similar to what the traits are. They don't respond to organism, but they respond to what carbon, nitrogen, or phosphorus ratio these plant litters have. That's what the driving factors is, traits. Origin is not a trait. So let's get to the systems, what we were thinking about. The problem that we always facing, and Andrea communicated about that, is how do you assess an effect of, this is for deer, effect impacts in a landscape where you don't have true replicates, right? Every little piece of forest, every little landowner does things different. Every little piece of land has a different land use history. So how can you then assess what's going on and you can standardize what you're doing? We did this initially when I started working with deer, and I wanted an assessment. How many deer are there? What are they doing when we're working at West Point? So we developed just a direct approach of looking at how oak seedlings are being affected by it. And we validated that method. It took almost 10 years to make sure that we actually got it right. We had the wonderful opportunity to have deer management here on Cornell campus, and we knew that after many years of doing this, that the number of deer was a direct, or the proportion of oak sprouts was a function of the number of deer that were in a certain area. We had marked deer, we know how many deer we had. So here we know that what we were measuring, the response directly to the stressor, we were interested in deer in response to that. So we could validate our method. We tried something else. This is John Mertz and Vicky Nuzo again in Japanese knotweed, right? So this is foraging success. You see a little green frog, but it's actually not that little. We're repeating this in some other systems, but it's a bucket that we have a bucket transect through a Japanese knotweed into an old field situation. That border is here, and you can kind of see for plants, as you're getting into Japanese knotweed, your diversity of, this is percent cover goes down to very, very little, while Japanese knotweed cover goes up. The foraging success of frogs measured as the amount of biomass gained over a 38-hour period goes down the further you are in Japanese knotweed. It's a function of prey availability, right? They eat everything that they can get their mouths around. And here we have an impact of Japanese knotweed that we don't like in terms of amphibian performance. We also did our wonderful thing of going out and doing your meter square, and then you follow it for a long time, the prescriptive thing that plant community people do. We did that for loosestrife. Vicky and I did that at many different locations. And yes, we can find that over time, if you have a very long time, if you are allowed to do this, if you can maintain your sites for 20 or more years, you can see a change in plant communities, but it may take a very, very long time. So this is for biological control. So we keep on doing things. This is not something quick. This is not what people typically do. We're exploring other linkages between salamanders and deer. We now know there is a linkage. I'm not telling you what the data are, but if you exclude deer, salamanders are doing better. Surprising. We do things with plants, with amphibian and survival growth at Montezuma after Phragmites management. This is the bucket situation that we do and sentinel planting that we do. So we try to advance this management further. But what we need, as Andrea said, we need this established linkage between something that we think is a stress here to introduce plant and the performance of something else, or soil carbon, or whatever you want to do. We need to consider the feasibility versus the logistics and the monetary expenditure that you have, right? Somebody needs to fund this. If you want to do plant community surveys and want to do it for a long time, you need to have the botanists that are able to do that. But ideally, we would like to have a rapid response, so we gain information very quickly. We'd like to standardize the approach, and that's not necessarily the species. We're trying to standardize an approach on how to do the assessment, right? People in Long Island or in the Adirondacks or in the Finger Lakes region may deal with very different species communities, right? I'm not telling you, you need to pick these particular species. Red oak was a good one for deer because it's so widely distributed. So we're thinking about portfolio. We've done some other things. You have read about, if you read what was sent to you about Audrey's approach to look at management of Phragmites in the Adirondacks, or Brendan's approach and how to fine tune the approach about deer management and not just accessing red oaks, but also other herbaceous species. And not everything needs or should be measured or even can be measured. Which ones we are selecting is really going to be important. We're going to show you some examples now, but I want you to do this stuff so we collectively learn more. I understand that you are short on time, expertise, and people, but there has to be a mandate. Otherwise, we're not learning from each other. You do so much out there in the field, yet other than a meeting like this or talking to each other, this is my experience. If you don't quantify what you do, you're not going to learn much. So we will do more. We will hear more about some approaches, how we have tried to do that. There's more work to be done, but it's not something that's very quick.
So we can talk to Bernd at the break, but I'm going to bring up Hannah Kirkland to get started with her presentation as we start delving into some examples of what this might look like.
Hi, everyone. Sorry, I'm just trying to load my notes. So I'll be talking today about my work adapting the sentinel method that Bernd and others have been working on in the US, and I'm using them to answer similar questions in Scotland, specifically looking at impacts of deer and the impacts of vegetation and how we're managing that vegetation on the recovery of woodlands. So I'll start off with some background. So I don't know if anybody's been to Scotland or even if you have, you probably aren't familiar maybe with our uniquely concentrated pattern of land ownership. So we have huge areas of land under private ownership, and it's in the hands of a relatively small number of people. And they have exclusive rights to hunt deer on their properties. So what this means is that property owners, land managers are running these large properties as what we call deer stalking estates or deer hunting estates, where paying clients can pay large amounts of money to come and shoot deer. And on these properties, deer numbers are maximized to try to maximize hunting opportunities. And the industry is centered on these red deer, which are, I guess, a cousin of elk, though a lot smaller. But we do also have other species of deer, including our native roe deer and non-native species like sika deer. So huge areas of our countryside are dedicated to these stalking estates. And at the same time, very little of our landscapes are covered in forest. We have about 4% of the countryside that is native woodlands. And although the reason that we have so little, they're varied and they go back centuries. Today, it's our large deer populations that are the big concern. Overbrowsing are preventing regeneration and causing further declines. So the Scottish government and conservationists are calling for huge deer culls. So large, we need to shoot a large number of deer in order to enable our ecosystems, which also include peatlands, from recovering. But it is worth pointing out that it's a voluntary system of deer management, so the government don't actually have any legal powers, and landowners can shoot how many they want. So, deer culling is going to be a massively important part of the solution if we want our woodlands to recover. But evidence suggests shooting more deer doesn't always guarantee a recovery. And even those places that are carrying out deer culls, managers don't really know if what they're doing is working. They're making decisions largely based on how many deer they shoot, how many deer they think on the landscape, are on the landscape, and they can visually try to see, okay, do our woodlands look like they're improving, but they don't really have much data to evaluate the outcomes of what they're doing. And secondly, even if the deer reductions are working, there are potentially other obstacles that need to be overcome to enable our woodlands to recover because they've been decimated for centuries. There's legacy effects. And so a big question remains, what else do land managers need to do beyond deer reductions to restore woodlands? So I don't believe these questions can be answered using the assessment methods that are currently used across Scotland and the UK in general. There are a few of them, but probably the gold standard is what is called the Woodland Herbivore Impact Assessment, and it's the one that is proposed by the government. I think there are several important limitations that Bernd and Andrea have kind of already hinted at. Firstly, to carry out these methods, you have to read a 53-page guide. It's really complicated, it's confusing. I don't fully understand how people are using these methods. But it is basically, you have these indicators and surveyors are assessing impacts through observations from no impact to very high impact. So a recent review of the methods showed that surveyors are coming up with very different results when they're surveying the same piece of land because it's based on observations, not measurements. So the reliability of the data these methods produce is unclear to me, or potentially very poor. Also, I think we miss a lot of the data with these methods. I don't think they capture the true extent of impacts because often our woodlands are so degraded, you can kind of get a sense maybe in this picture here. Often we just have no saplings in the understory. They're so excessively browsed, in which case surveyors record it as not applicable rather than evidence of high impacts. And secondly, relying on existing vegetation and evidence of recovery is obviously a slow process, and managers can't really use this to quickly evaluate whether their deer reductions are working. So that is why I was interested in using the sentinel method and adapting it to a Scottish context. I don't know how many people are familiar with the setup or what it actually involves, but I guess we'll go through that later on today. But I'm essentially using an identical setup. So I'm introducing these plants into the system, often that aren't there in the field. I'm measuring things like survival, height, whether the plant has been browsed or not, and I'm also recording other stresses as well, which in this system, we don't have a lot of, I mean, in the UK, we do have invasive species, but up where I work in Scotland, it's not really, well, it doesn't seem to be that relevant to my woodlands in particular, but things like rodents and invertebrates are also putting plants under pressure. But crucially, I'm obviously working with different species to the ones that Bernd works with, and I'm using this portfolio that he mentioned. So in total, I'm actually working with 11 different species, including an oak, but also aspen, birch, and a bunch of herbaceous species as well. So last year I set up my first experiment using the sentinel method to get at this question of whether deer culling that managers are undertaking is improving the performance of woodland plants and reducing browse rates. So I selected 12 study sites where managers are attempting to reduce deer numbers. I'm using 6 different species. 5 of them are woody. 1 of them is a herbaceous plant, which is our native goldenrod. And for each species, I plant 20 individuals along a transect, and the species are in an alternating pattern. They're about probably 5 meters in between each plant of the same species. Almost 1,500 plants for this experiment. And I monitored them for a year. So I finished collecting data on these plants this spring. And the idea is that the data I collected during that year serves as a baseline. And I'll return in 2025 next year to plant a new cohort of seedlings to detect whether browse rates have declined in that time. I predict that they won't, that more sustained deer culling will be needed. So the idea is for this experiment to be a much longer term experiment that could potentially continue after my PhD as well. So the second experiment I'm working on has to do with whether managers can be doing other restoration activities, land management activities to help facilitate our woodlands to recover. For this experiment, I'm working at 3 sites, and I have 3 plots at each site, so 9 plots in total, and each plot corresponds to, so I burned, I set fires, I mowed the vegetation, and there was an experimental control plot where I did nothing. And the idea was to get at whether these treatments, and this is a heathland system, which are common in these deer stalking estates. So whether managing this kind of dense blanket of heather could help our woodlands recover as well. Using 3 species for this experiment, again, 20 individuals per species, but 20 caged and 20 uncaged, and caging them means that I can also test how these treatments work in the presence and absence of deer. So these experiments are in the early stages, and I don't have, I haven't analyzed the data yet, but so far they're working well. And I just thought I'd run through some of the challenges that I had adapting these methods to Scotland. So we started off with the idea, I know Bernd grows his own plants. And I was originally planning to do that, but quickly realized with the quantity that I had to grow and the fact that I didn't have Cornell's facilities, I wasn't going to be able to do that. So to carry out both these experiments, we just purchased plants from nurseries, and that worked really well. They're being browsed, they're surviving when they're planted, and it was relatively cheap and saved me a lot of time. Another challenge was choosing the species that I was going to work with. I think my sense is that in the UK, we just don't seem to have the data that you guys have in the US. So I didn't have a lot of data to rely on to choose my sentinels beforehand, which is one of the reasons why I'm working with so many, so that I can find out for myself through these experiments. And this was particularly the case for the herbaceous plants that I wanted to work with. There was almost no data for any of them. And then the other challenge I had was that the methods that were devised here, my system is very different and the landscape is incredibly different. But the whole idea is that these methods are adaptable. And so all we needed to do was a few adjustments and it's working. So I think one of the key points to mention is, so I think in the original protocol, you know, you want to avoid places where deer don't browse, like steep slopes, you know, right next to big boulders and things, places where deer won't browse. And I was trying to do that at the beginning, but quickly realized that's exactly where our woodlands are growing. So if I wanted to do this, I just had to go and plant them there anyway. But crucially, the deer don't have a choice either. Because we have so little woodlands, if they want to come and browse our saplings, then this is where they need to come and eat. So despite the fact that I'm planting in really steep ground and right next to boulders, the plants are definitely still being browsed. So that was quite a big challenge, but just a couple adjustments made it work. And then the last point I wanted to mention, which Bernd reminded me of yesterday, because I think I blocked them out, was the Highland cows that I had to deal with. So I tried to avoid sites with domestic herbivores, but we have a lot of them as well. So I couldn't avoid these cows. And they were a really big problem for my cages. They probably added on days of field work, checking the cages, repairing the cages. And I never really found a solution other than just doing that. But that's something I wanted to mention, you know, if you're working with cages, probably avoid places with cows. They're so inquisitive and they like to scratch themselves up against the cages. So that's my work with the sentinel method in Scotland. This is one of my study sites. Pretty beautiful landscapes, but incredibly degraded. And so it's really important that our managers, those who are trying to manage deer sustainably and restore our woodlands, have the data they need to make good management decisions.
So we're going to move on because we have just one more talk before the break. And then, so if you do have any questions, I hope we can hold on to them for later, but we're going to have Lainie Brice speak next.
All right, good morning, everyone. So just a little bit more before you get a breather. So I'm going to talk today about my work using the sentinel method, and it's a little bit different than maybe what a lot of you are used to. We're taking the sentinel method to expand beyond looking at just the effects of deer on forest regeneration, but the effect of predators on forest regeneration. So the main goal of this project is to assess whether the presence of wolves in particular can reverse negative deer impacts on forest regeneration. So this is our beautiful white-tailed deer. I'm sure you're all very familiar with it. Typically, we've been using the sentinel method to look at deer impacts on forests through browsing. And we're extending this to look at how wolves might affect forests by their direct effects on killing deer, reducing the population, or scaring them away. So essentially, can wolves reduce local deer density in a way that will benefit forests? This is a trophic cascade. It's the classic example of wolves ungulate plant. I'm sure many of you know the Yellowstone story. Pretty famous, pretty touted that wolves were reintroduced in the 90s, drastically reduced the elk population, allowed all of the riparian vegetation, upland aspen forest to come back. I actually spent my dissertation in that system working on this very question. And it all comes down to, we don't actually know how to measure those kinds of effects. So the story is very well publicized, but the uncertainty around it is massive. So my work essentially suggests that the wolf effect is much, much weaker than what we know. But the reason that we don't really know or didn't really know is because methodology is hard. So today I'm going to talk about how we try to get to this question using a slightly improved metric. So this project stems from the challenges we've had managing deer in New York. Bernd has done a lot of work with this. Different hunting methods aren't necessarily helping. So then the question becomes, well, just bringing back a native predator, will that help? So to do that, we're actually hopping over to Wisconsin. Wisconsin has very similar forests. They're all classed as, well, in the north it's classed as northern hardwood forests. The understories have similar species. And we're going to Wisconsin because crucially, they have wolves. So I'm going to give you a little bit of background here. So this is the upper half of Wisconsin. Wolves are native to Wisconsin. They were thought to be eradicated around the 1950s, but then they started naturally recolonizing from Minnesota in the late 1970s to early 1980s. So where's that stick? So, okay, so this is Minnesota over here. And so the first wolf packs kind of started coming back in over here. So each of these blobs is a wolf pack. You can see that there are these two here, kind of suggesting that wolves never really actually fully left. It's hard to measure these kinds of things, but in the 80s, we had just kind of a few packs that are coming in, starting in the Northwest. And then this is the most recent map where each of these circles is an estimated wolf pack. And so you can really see that the wolf population kind of started here and was spread out. Each of these wolf packs is only about 3 to 4 individuals. It's just basically a family group, very small. Most of the wolves are in this northern section, and there's a very small amount in central Wisconsin. But you can see that the population has really grown over the last couple of decades. And just to further point that out, this is a graph of wolf abundance is the red line. The blue line is the number of wolf packs. So starting in 2000, we started with about 250 wolves in 2000. The last couple of years, it's kind of hovered around 1,900. You see that dip from 2020 to 2022. That is from a wolf hunt in the state, and wolf hunting goes back and forth with whether the wolf is listed as endangered or not. So that's why you see that little dip there. But in recent years, we have 900 to 1,000 wolves in Wisconsin in close to 300 packs. So if we then jump to deer, which is their preferred prey, the data on their population is much shakier. It's very hard to estimate deer abundance, but we can kind of take away from this. This graph is estimated deer abundance from 2006 to 2022. It's estimated from hunting data, which is biased in and of itself. So hard to trust, but this is for the entire state, and it's estimated that there's somewhere around 1.5 million deer across all of Wisconsin. So like here, big deer populations, and managers are worried about impacts on forests. So what we decided to do was use the sentinel approach to try and address this. So what we're doing is we plant and monitor oak and goldenrod seedlings and measure browsing. Critically, the way this is different from what we do here is that we're measuring it across a gradient of wolves. So this is a map of wolf density in the state. It's based on a wolf occupancy model, essentially. All of the pink stars are our study sites. So we have 40 sites across northern Wisconsin. The crucial part to this is to really get a gradient of wolf density. So the darkest hexagons are the most wolves. It's only about 2.7 wolves per 100 square kilometers. So it doesn't sound like very many, but that's what the highest density is. And we really tried to make sure that our sites spanned this gradient evenly. So sites with no wolves, that's the white, sites with lots of wolves, and then everywhere in between. And similarly, we are also looking at wolf occupancy time. So this is basically wolf packs over time stacked on top of each other. The dark blue is where wolves have been for a really long time. Light blue is where a pack has only been for a few years. The idea here being that where wolves have been for longer, they should have stronger effects on the deer population. We would expect to see less browsing where there are more wolves and where they've been for longer. So what we did in our first year was we grew thousands of oak seedlings. These are acorns that came from Wisconsin. So we had someone collect them for us, ship them here. And then in our greenhouses here on Cornell's campus, we planted them in these cones, grew them. And then in June 2022, our first field season, we drove them 2 days out from Ithaca and we started planting. So you'll see this in the field this afternoon, but essentially we have train set tape, the yellow line, says Bernd with his drill. Someone goes along with the drill, drills all the holes, someone follows behind with all the plants, and we planted them all out. So the first year we planted both red oak and white oak. So here in Ithaca, we mostly used, and in New York, we mostly used red oak. We decided to use white oak as well. It's common in southern Wisconsin, not as common in the north, but it's thought to be moving north with climate change. So we wanted to see if that was a useful sentinel. Our goal was to plant 20 individuals per site. Growing the white oaks turned out to be a little bit harder than we expected, so they were pretty small, and we ended up kind of axing a bunch of them. They were just too little. So you can see, so here we really only planted at a subset of sites with the white oaks, and really cut that sample size down. But we ended up with about 1,100 plants that first season. You can see the red oaks on average about 14 centimeters tall. The white oaks only about 11 centimeters tall, so a little bit shorter. But that was our first field season. So we planted all those in June. And then in August of 2022, so late July, early August, so about 2 months later, we went back out and we measured all of them. So we measured a lot of things, but today I'm really just going to talk about deer browsing, but we also looked for rodents, rodent damage, leaf damage. You know, we measured surrounding vegetation, lots of different things. But we went out, we measured all of them again. And our first year, less than 5% of our plants were browsed. So this was way lower than we expected. When we plant them in New York, they're eaten up pretty quickly. And less than half of our sites even had deer browsing. So this was disappointing. We, spent a lot of time growing them, putting them out there, and didn't quite know what to make of it. Our first thought was that the oaks were maybe a little bit too short, and deer just weren't, bending over that far to eat them. But what we really thought was that the understories in Wisconsin, even though on paper they sound like they're similar to here, they just had a lot more going on in them. They looked a lot more flush. There were, this is, a carpet of trilliums that we came across May 2023. So deer just had more options of things to eat. Oak is, they'll eat oak, but it's not their favorite food. It's one of the reasons that it's chosen as essential, because you want something that they'll browse, but they won't eat all of, so you can actually try and get a gradient of browsing. But so it seemed like they just had other stuff to eat. They weren't really all that interested in the oaks, or they just didn't have to rely on the oaks. So for our second year, we said, well, how are we going to adapt this? How are we actually going to get some usable data out of it? So there are 3 different things that we were kind of thinking about. So first was to plant taller oak seedlings. We thought if we can get them taller, if we can get them to like 30 centimeters, they'll be closer to kind of the preferred browsing range of deer. They'll be maybe taller than some of the understory vegetation. They'll stick out more. The second thing was to try and get a longer browsing season. So when they're planted in New York, deer tend to eat them pretty quickly. So, a couple days later, 30 days later, they're eaten. So here we thought, okay, if we plant them earlier in the season and we go out to measure them a little later in the season, they'll have more time to be browsed across the summer. And then the last one was to try a different sentinel, maybe try a species that they like more, that'll be more appealing to them. Maybe we'll get better browse rates. So first of all, we tried growing our oaks taller. We planted them earlier in the year. They did not get any taller. They probably put, the resources into the roots, but didn't really pan out. So that one was axed, but we did have a longer browsing season, and we tried Solidago plexicalis. So I'm just going to call this goldenrod. It's zigzag goldenrod. But we tried an herbaceous species that we thought they might like a bit more, and it grows taller than the oaks more quickly. So we thought we would give this one a try. So in May 2023, we repeated this. I should note logistics, in case you're thinking of doing this. It's a real challenge to grow plants, in one state and get them to another. So instead of driving them out this time, we tried shipping them with FedEx. It was real stress. If you have questions about it, we can talk about it later. But so in May 2023, we went back and we planted 3 species. So we stuck with our red and white oak. We wanted to see, give it another try. And then we also planted goldenrod. So in this case, we were able to plant 20 of each species at pretty much all of our sites. The red oak and white oak were about 12 centimeters tall. So the red oak actually shrunk a bit from the previous year, opposite to what we wanted to do. And then the goldenrod, though, was almost 20 centimeters average when planted. So we did get a little bit of a height boost there. But so all in all, this was close to, close to 3,000 plants, close to 4,000 across both years. So a pretty massive sample size across quite a few sites. So this is the browse data from that second year. So for the white and red oaks that we planted that second year, still very, very low browse rates. Only 1 to 2% of the stems were browsed. Again, not great. But goldenrod, 31% of the stems, so like of all the stems were browsed. And we had a good range of browsing across sites. So this histogram is just showing how many sites had a certain amount of browsing going on. And you see we get kind of pretty much full coverage from like very little browsing, under 20% of stems were browsed at a site, to upwards of 80%, 90% of stems were browsed at a site. And this is the gradient that we really need in order to actually do any kind of analysis. We need that variation. So we were very pleased that goldenrod seemed to be working out. And this is after 3 months of, being in the ground, 2.5 months of being in the ground. And just to highlight that difference, this is that browse probability model accounting for variation and stuff. So we have our white oak and red oak, and you can see, that there's that 1 to 2% browse probability, and goldenrod is 10 to 20 times more likely to be browsed. But we didn't forget about the oaks from the first year. We measured them too, and just want to show you this as an example of how the browsing has kind of increased over time. So this is, we planted June 2022. The gray dots are just white oak and the red is red oak. And so we start off, this is our first visit back in like July, August 2022, very little browsing. We went again in September that following year thinking maybe they just needed more time. Maybe we would get higher browsing. A little bit, but not really. Then we jump a couple of months, almost a full year, to May 2023. You can see that browsing didn't really increase a ton. So we didn't seem to miss a ton of browsing over that winter and spring period. And then here in August 2023, we finally see a little bit more of a jump. So we figure that maybe once the oaks had been on the ground for a full year, their leaf out matches up with everything else. Maybe now they can be a little bit of a better indicator. So we did get, after a year and change, we did get some usable data out of the original oaks that we planted. And to compare this back to New York, this is essentially a similar graph where we have cumulative browse. The red is red oaks in Wisconsin, the blue is red oaks in New York. And you can see, so this is essentially what I just showed with the red oaks over time. Very little browsing happens, and then after 400 plus days, we get to about 15-20% browse probability. In New York, you see that the oaks are browsed pretty dang quickly. So after, what is this, like a month, it's like 50% of stems being browsed. After 3 months, we get to like 70%. And then after a year, you're up to 80-90% browse probability. So red oak works really well as a sentinel in the forest that we planted it here. In Wisconsin, not really. So I just went on about all the challenges we had with this method, but our adaptations worked. We did get good data out of it. So this circles back to our actual question: is there a wolf effect? So we expect, right, where there are more wolves, they're going to decrease the deer population or scare them away. That should mean that there's less browsing in the system, and that would be what we consider to be a trophic cascade. So I'm going to show you just the browsing data from the goldenrod, and then the oaks from the original transect. So it's going to be basically a year and a half of browse data for the oaks and just 3 months for the goldenrod. These are models that include lots of different things, but I'm really just gonna show you the wolf effect for now. So this is our model, our final model of browse probability as a function of wolves. So this is wolf density. So here's high wolf density, here's low wolf density. The lines are the different species. So here's goldenrod and yellow, and then the red and white oak are, you know, blue and red. And we do see that as wolf density increases, browse probability decreases. And this is the exact pattern that you're looking for if you're expecting that wolves can cause a trophic cascade in this way. We also modeled it with wolf occupancy time, and we see the same pattern. So as wolves have been there for longer, you get less browsing. The challenge here is that browsing is plant damage, right? It doesn't necessarily, you know, correlate exactly with plant population growth or plant population response. So whether this, you know, decrease in browsing is strong enough to constitute, you know, a big flush of regeneration where there are more wolves and where wolves have been for longer, we don't quite know yet. With goldenrod, we still have, you know, 20% browse probability at high wolf density, so that's still a pretty high number. So that's a caveat here is whether that's actually gonna result in, you know, forest regeneration, but this is the first kind of linkage to show that, yeah, where we have more wolves, we do have a slightly decreased browse rate. So the key takeaways from this, goldenrod was a much better browse indicator than oaks in our northern Wisconsin forests. And the sentinel method, after our adaptations, after testing things out, it was able to capture browse rates in Wisconsin in a way that we could actually use and learn from. For the actual study, like I said, we have a wolf effect on deer browse that is consistent with a trophic cascade, but it's not yet clear whether the decrease in browse that we saw with wolves is going to translate or does translate to forest regeneration. But this is just an example of how we've adapted the sentinel method to kind of expand beyond deer, expand beyond invasive species, and look at a more broad community level question. So lots of people came to the field with me. These are some deer in Wisconsin. And I don't know if we are doing questions now or if it's break time. If there are any questions, I can take a few questions. One question. Go for it.
What was the overall difference between average deer density in New York versus Wisconsin?
That, you know, do you know, Bernd?
So how do you want to have it per square mile?
Sure.
I have no clue, nobody has. So we know the roughest estimate of, and I don't even know what the size, the state comparison size is, right? 1.2 million deer, best estimate of Brandon right now, right? In New York, 1.5 in Wisconsin, million deer. What that translate in per square mile, I don't know, but it's way beyond 20. Just to give you some kind of a feel, right? So this is not, these are not low deer population.
Yeah.
I've never seen as many deer on the side of the road as in northern Wisconsin. You don't see that in New York.
And that also highlights the challenge of getting at any kind of predator effect or even deer effect. We have a really hard time estimating white-tailed deer abundance locally. And so using the sentinel method, looking at browse rates, essentially it doesn't really matter how many deer there are, it matters what their impacts are, right? So that's what we're trying to use that method to kind of get around not knowing how many deer there actually are. Real quick, yeah.
Yeah, do you have any idea about why there was a lot more options in Wisconsin for deer browse compared to New York, of course?
I don't know, I'm not the plant person. This might be a better question from Bernd again, but I mean, their whole upper north is forestry operations. I don't know if that, or just less popular, it's very, like, not very populated up in north.
They're lucky. In terms of multiple sense, right? If you think about deer impact, I think, so it's my hypothesis, nothing really fundamental, but in terms of data, I think the impacts of deer have slowly moved west. It's still not recognized as much how bad it is, like in Iowa or in Wisconsin or even Missouri. And I don't know why that is the case, but the people in Wisconsin complaining about the heavy deer impact that they have in their woods. Yet when we travel out there, we saw, oh my God, as soon as we set foot in there, said our oaks are too short. Right? Because they were just blending in. So they were camouflaged by trillions. So that one is not the experience that we have here. You go in the woods, you plant an oak, that can be found very easily in most places in New York, right? in Wisconsin, even though they're complaining about the deterioration of the old woodland fauna, that's not the case. So that is a challenge that we need to leave. Once we step foot into it, it's like, oh damn.
So maybe like that historic, like wolf.
I don't know whether it's a historical, you don't have the full story, wait for the full story, at some point Lainie will tell you the full story. And so, yeah, there's no good explanation why that is the case, right? So maybe because northern Wisconsin has very little agriculture, there's forestry operation, that's fundamentally subsidizing the deer by clear cutting. And then so they're trying to manage the deer population by cutting by experimenting with the size of the cutting operations, because the flush of new seedlings, that's what deer food is, right? They go after that young regeneration. So, yeah.
So if you have more questions, we do want to take a break, so you can get up and stretch your legs before our last two talks and some broader discussion. So we'll reconvene at 11:30.
All right, everyone, welcome back. Welcome to the first day of the semester here at Cornell. What we're going to do now is we're going to move from the uplands and from the terrestrial systems, and we're going to talk a little bit about how we're taking some of these concepts and applying them in an aquatic context. Now, I work with water chestnut. This is a water chestnut bed on the Seneca River. And I'm sure many of you are very aware about water chestnut, but it is this prolific, very impressive aquatic invasive plant that is rooted in the sediment, and it produces these floating leaves of vegetation that can come and dominate large areas of open water. Now, this plant is the target of management, most conspicuously because of its impacts to recreation. It is very difficult to boat through. You hear people grumbling about not being able to fish in it. We might have some shifting going on between presentations here, but this will make it work. So one thing that we do know about impacts of water chestnut is that there are some big impacts potentially to water chemistry, mostly to oxygen and to nitrogen. Now, this is just showing how low some of these oxygen values can get in water chestnut beds in the summer and the autumn and some Mohawk River sites that we've been working on. Now, below this 2 milligrams per liter level of oxygen is classified as hypoxia, which sensitive aquatic organisms have a hard time surviving in. So this is one of the impacts we know about water chestnut. And it's likely that these impacts to the chemistry are going to be explanatory or at least helpful in understanding any of the impacts to the ecology of these systems. Now, there are a lot of suggested impacts, but actual experimentation of how organisms survive in these water chestnut beds is pretty undocumented. And so that is what is the basis of the summer project that I am just coming out of. So this will be some initial results from that. So this is a challenging system to work in. Not only is it extremely dense at the water surface, this is a rooted plant that has plant parts throughout the water column, so it's very hard to navigate through. And in addition to getting out into these beds to take data, you're also working on the water, right? So you have additional logistics of boats and access and needing to get out there. Now, another complicating thing is that we've never been able to replicate both the plant productivity or the chemistry conditions of in-the-field water chestnut beds in the lab. So this gets back to, can we try to test some of these questions that we have about the ecologies of these systems in the lab? I'm not sure I would believe those results. So the general background of what Lainie was talking about and Hannah in Scotland is this indicator or sentinel approach, which basically boils down to exposing species to environmental conditions of interest, whether that's wolves or deer abundance in Scotland or in and out of an invasive plant, and then monitoring their performance over time. And so this is the approach we wanted to take for the water chestnut bed system. But how do you test a plant species in this system where it is hard to get to? We don't have too many floating aquatic plants here in New York anyway, so you're limited in floating plants and tried to imagine planting something in that sediment and coming back and monitoring it over time. I'm a plant guy, I wanted to do the plant stuff, but we decided, well, we're gonna need to switch here. And let's think more about some animal models or organisms that would be likely to occurring in these systems. Well, that quickly presented another challenge of, okay, we need to come back and monitor these individuals over time. How do you do that? And so what this involved was creation of these deployable cages in which we monitored organisms over time. So let me give you a little bit more background about what we did. Okay, some formatting stuff. So we wanted to work with at least two different taxonomic groups here. So we chose a fish species and we chose an amphibian. We chose this fathead minnow, which is, I learned, the most widely distributed bait species in North America. It is a generalist, it's a tolerant species, and it's widespread in New York watersheds. The nice thing about this is that there are certified New York State hatcheries that you can go and purchase these fish from, certified as disease-free. So we wanted to work with, this felt like a compelling organism. Another one that the Blossey Lab has a lot of experience with are with using tadpoles of various frog species. And we selected green frogs, which are another widespread amphibian commonly found in wetlands. And these were collected as egg masses from our near research sites and reared in Ithaca. Now, these were not tiny little newly hatched tadpoles, but rather large enough to have developed some lungs and to be able to gulp air from the water surface. Now these, I've never seen a tadpole in a water chestnut beds, but I've never seen more frogs on water chestnuts than anywhere else. I can see some fish swimming nearby on the edge of water chestnut beds, but it's pretty uncertain what's going on in the fish community. So these felt like two great starting organisms to try to test these protocols with. And so what did we do? We ended up going through multiple designs of cages to try to figure out how to best create a hospitable environment for these amphibians and fish. And so what we ended up doing was creating these 12 by 12 by 14 cages that we made out of welded wire and then wrapped again with window screening to prevent anything from getting in or organisms getting out. And importantly, they float with these nice, very scientific pool noodles here. And the other benefit of this design is that there's this protected access to the water surface, right? So these tadpoles or these fish could come up to the surface safely and not get nailed by something within their mesocosm or in the cage. Okay, so we needed some place to put these cages out into the environment. So the biggest regional infestation of water chestnut in this area is at the north end of the lakes in the Erie Canal and the Seneca River system. And that's where we went to target our systems. So we wanted sites that had large trappa beds, where water chestnut is trappa. And then we wanted some reference comparison area without water chestnut. Ideally, we wanted something like this, so a white water lily or a yellow lily, something comparable. But we were only able to find that in two systems. But most importantly was we wanted it embedded in a big trappa bed and adjacent to with no travel. We needed some easy boat access, and importantly, this required a lot of permitting, so we wanted to consolidate all those logistics and just work within one DEC region that was regional for us and easy to access. So we worked along the Seneca River. This is Howland Island. There's massive water chestnut bed all the way around this island here. This is another little island, and then Cross Lake has a big embayment with a large water chestnut bed that you can see from these satellite photos. At each of these sites, what we did was we took 8 of these cages, we put them deep into the water chestnut bed, not deep, but we put them interior water chestnut bed, and then we put 8 outside of the bed. After the cages were in, we stocked them with our organisms, and then for a 5-day period, and every single day I came out and checked them for survival and fed them. We tested the minnows first, 10 minnows per cage. And then after that assessment was over, I removed them, and then we added the green frog tadpoles at a lower density, 4 individuals per cage. Okay, so this is just what some of these sites look like and getting the cages out there. And then these were the data that we were collecting, survival, plant coverage, dissolved oxygen, water temperature, depth, and some other water quality parameters. Okay, so these are raw data, early results. I'm just coming off of this work. So I just want to explain what these graphs are going to look like over the next series. So we got day on the x-axis and survival on the y. We're going to have two different lines. The gray is without the water chestnut, the reference site, the control site, and then that orange color is going to be within the water chestnut bed. So we're going to start with the minnow survival first. Well, this is kind of the trend that you would hypothesize, or maybe society would hypothesize about the impacts of a lot of invasive species, right? Here we have a clear and pretty immediate reduction in survival of these fathead minnows. However, and another important thing is showing that, the control worked too. There were some deaths in these reference sites. However, the vast majority survived. And so our cages worked. Some fish were happier than others. These in the deepest part of the water chestnut beds had very low survival. On the edges, it was a slightly different story, but here there's a clear signal of this invasive plant having a negative impact. Now, what about the tadpoles? Now, the tadpole story is a little different. We have much higher survival, or higher survival for the tadpoles, but lower overall in the reference. So we still have a slight decline. They didn't do as well in these mesocosms as the minnows did, but still a pretty clear difference between the within the trappa bed and outside of the water chestnut bed. Now, I told you in the beginning that water, dissolved oxygen in these water chestnut beds can be extremely low. And that is what we are showing here with this value cell on the left-hand side down here. Now, here's survival. This isn't in percentage, but 10 would be 100%. So down here in the lower left, this would be low oxygen, low survival. That's kind of what you would expect. However, there's a lot of higher survival in these really extreme oxygen regions. So it's something in addition to, or more than just oxygen. Note large range in survival. So we have these three sites, and there are large site differences between these different water chestnut beds. So here at this, Bonta Bridge was the most inhospitable for our organisms that we tested. Now, why might that be? The oxygen wasn't much lower, and in some cases was higher than some of the other water chestnut values, but it was way more lethal. I'm ahead of myself. Well, so we don't have the answer for that right now. But there are a few things that could be suggestive. So this was a warm, shallow embayment. There was minimal water exchange, apparently, with the Seneca River. And so maybe it's something to do with the residence time of the water in these water chestnut beds, not exchanging much with the much larger volume of the Seneca River, which quickly dilutes that low oxygen system. And so it's unclear what combination of factors led to this. However, a big takeaway from this is not all trappa beds are created or are equally lethal for these organisms. So just some conclusions here. So these indicators do reveal the stressful conditions of some trappa beds. However, like I just said, there seems to be a lot of site variation and some factors within those sites that are not yet explained that are important to these survival trends. We find that fathead minnows, surprisingly, were a lot less tolerant of the trappa conditions than the green frog tadpoles. And this could be possibly just due to differences in physiology, and the tadpoles are more effective at gulping air from the surface. They come up to the surface with the surface tension, grab an air bubble and take it down and consume the oxygen from that, which fathead minnows might not be able to do. The DO appears important here, but there are other factors at play. This could be, so whenever you get oxygen severely depleted from a system, you can get some crazy changes in water chemistry, right? If oxygen's not accepting those electrons, something else is. This also could be a place where pathogens are accumulating. So this was a trial test here. Can we use animal indicators here in these beds? And what can we learn from them? Well, one thing that we learned pretty quick is that research involving animals adds a lot of complications. It's very time intensive for all the husbandry involved. It's very permit intensive because you cannot just place tadpoles or fish anywhere. You need permits for all of that, of course. And you know, there's also this complicating thing in river systems where I was working where couldn't they just leave? Couldn't these organisms just leave? And to that, maybe. However, some of these systems, water chestnut is a major habitat type. Many of these wetland areas that would have a diverse plant community have now just been replaced with water chestnut. So it's a very major part of these watersheds. And water chestnut just doesn't just invade flowing waters, it invades ponds and still waters too. So in those systems, these organisms would not be able to escape these signals. And so another challenge here with these cages and deploying animal models is there's nothing mechanistic about this, right? We're kind of telling a story about oxygen because I think that's going to be really important in explaining these survival trends, even if it's indirect. But this is more outcome oriented. This was like an exposure test. Are these systems actually as harsh as we think they could be, or are there ways for these organisms to circumvent these harsh harmful conditions? So I think that the most potential for the system and the deployment of these aquatic indicators are really pairing it with the management. This was a new friend of mine, Chuck, who was a landowner next to one of the areas I was working. He's developed this plow on top of this pontoon boat. He got his neighbor to make one too. And they are responsible for this open water around KTA Island. If they weren't doing that, it would be completely covered over. Now, I want to point out here that in these systems, I used his open areas that he had cleared the year before to place my cages. And he was able to, well, the result of the open water brought the oxygen up to 2 to 4 milligrams per liter. Within the water chestnut bed, just 10, 15 meters away, it was often less than 1, always less than 2. And if you just look at the difference in survival there from, 10% to over 90%, that's pretty astounding for just a simple management action here. So I think that applying this concept and pairing it with management and asking questions of can reductions in plant coverage lead to better outcomes for these species? How large of a reduction does this need to be to achieve these oxygen effects and the biological communities? And this is important. I work on the biocontrol system for this, so I'm thinking about how much of these rosettes are going to be consumed by the beetles. But this could also inform mechanical. Maybe we just need strips out of these large beds rather than full removal. I think this has a lot of potential for other systems, testing other indicator species, and I think I'll end with that. I think that was my last good thing to say. So thank you for the people making this work possible, and we'll take questions at the end, or?
Yeah, we'll take one. A quick question for Wade? Or I'm gonna dive right into mine.
Yeah, sounds good.
One question.
Yes.
You've probably looked at the numbers from the Hudson River data sets.
Yep.
They have exchanged twice a day, I believe, due
Yep.
to tides.
Yep.
How do your DO up and down the ultimate lows for both systems compare?
They bottom out at the low area, at low, similarly low values, around 1 or less. But that's just for a few hours, like you were saying, for the biological communities there. So you're getting that replenishment with tides. Ours are sustained, so we think that the dynamics are going to be pretty different, both for chemical cycling and also for the biology. But there's pretty cool adaptations of those communities there where they upregulate hemoglobin and ways to sustain through those low oxygen periods. Seems a little more stressful here, but to imply that these systems are ecological deserts is far from the truth. There's a lot of life in them. We just need to understand the mechanisms more.
Well, thanks, Wade.
Apparition, thank you.
So I'm going to now talk to you all about work that I actually did during my dissertation at the University of Missouri, St. Louis. And this is work that's another animal indicator, but working with invertebrates, ants in particular, and in a prescribed fire management system. So just to justify why we're using ants as a potential indicator group, ants provide a lot of different ecosystem services and functions, such as seed dispersal, but also other roles within the food web and soil nutrient cycling as well. But in addition to their ecological roles, ants also can respond quite similarly to other groups of interest, such as other invertebrates, soil microbial biomass, and birds when we're looking at abundance and diversity responses to disturbance. And so that's important because ants are easier to measure than a lot of those other groups. You don't have to deal with the permits to nearly as much of an extent because they're invertebrates. They're not very highly regulated. And it's fairly cheap and easy to sample ants as well. Really, the biggest time and expertise concern is just being able to process and identify ant samples. So when thinking about then this potential for ants as an indicator group, they've actually been tested and used as indicators of ecological recovery and resilience in different disturbed and restoration systems. A lot of this evidence is coming from Australia, particularly in mining contexts, but there are also other examples worldwide, including in North American grassland restoration contexts, of looking at how ants can provide a little lens into how broader responses are happening and can be easily measured. But that being said, if we want to think about using ants as a potential indicator for a new system, for a given system that you're interested in, it's important to think about, will they work in that particular context? And so we also know that there is variety in how ants respond, and it's not always in a predictable manner that might be really informative to management and to making conjectures about what is happening in these other groups. And I'm putting up there also that because this is going to be looking at a fire managed ecosystem, we know that globally in forest ecosystems, fire decreases ant diversity. So there's this potential for using ants then in a fire managed ecosystem, and specifically looking at fire in the Missouri Ozarks and other similar oak hickory forests throughout Eastern North America. Just some important baseline information is that there's an assumption of these forest types being adapted to some level of low intensity, but fairly frequent fire. There's a lot of human history behind that with Native American land use and then subsequent disruption with settlement colonization by Europeans. But current management practices in a lot of this ecosystem type now are returning to using some level of fire, whether that's annual or up to 10 to 15 year burn time between fire, depending on what your management goals are, depending on what your outcomes and your current starting baselines are as well. But that being said, there still is some that's fairly frequent for fire being applied to this ecosystem type in a management context. And so if we want to think about, well, how might that actually be having impacts on the ecosystem? Is it doing what we think it is doing? That's where I'm coming in with ants to see if there is a measurable response with ants and also if ants are just generally usable in this context. And so I'm working with two sites that are in Missouri. They're within the Ozark forest ecosystem type, but they're not too far from St. Louis. And so you'll note that this is coming from pandemic times to start with. So my first year I was working at a different site that had a little bit longer of a site history with fire from the past 20 years being managed to some extent with fairly frequent up to 3 years or even annual in some cases fire return intervals. My second site for the next year had a little bit longer of a site history without fire, just because it'd been governmentally owned for the military use prior to being acquired by a university. And so the first instance of fire on the areas where I was looking at Tyson Research Center is going to be in 2017. The next burn was in 2021. I do also just want to thank a lot of the field assistants that I had because they helped actually be able to get out and get information about what the ant communities were doing. And so just to look a little bit more close up at these two sites. So the first year in 2020, we had two burn sites that were fairly close together. Unfortunately, that's just how it happened with what was scheduled to burn and what actually got burned before shutdowns of COVID. And then we have two control sites that are a little further away. My second year had a little bit better spatial layout with burn sites being closer to control sites. And there's also an additional type of plot within there where we removed leaf litter. I'll talk about that a little bit more after I just start with comparison between burn sites and control sites. And the plots that I'm talking about here are 1 by 1 meter squares where we have pitfall traps. And so we used fairly small ones. They're little deli condiment cups. And we left these open for a night, 24 hours for the first year. And then we were aggregating responses in abundance and diversity from all of these to count as one plot. And so, again, thinking about these evidence showing that generally there are negative impacts of fire on ant communities and other systems, but also thinking that potentially if this is an area that might be better adapted to have fire happen, we wanted to see if fire actually is reducing ant abundance in these systems, first and foremost. And so in that first year, we sampled 3 different times, and we saw that for the first sample times, which were closer to when the burns happened in January and March, we didn't see any difference between abundance of ants in burned and control sites. We saw actually a higher abundance of ants in our burn sites in September, but that's also begs the question, was that due to fire? Was that due to other factors at that point? We also wanted to look at other aspects of the ant community, however, and so thinking about species richness and diversity, I used 3 different metrics of diversity, starting with just number of species that you have on the left, and then as you change the abundance of different species in that community, letting that be weighted to different extents with this middle metric, the Shannon metric being medium weighted, and then the last metric being extra weighted for just how abundant certain species are in that community. So we're getting a span of what species are there, but also what variety we have in abundances in these species. And so we wanted to look at all of these metrics. I'm going to walk you through them. And so we have these standardized curves for how the ant diversity was playing out in our different treatments and throughout the sampling time since burning. And you can see they're really clustered together, and it's really hard to sort them out. When we're looking at species richness, it's a little easier when we start weighting species by their abundances. But when you actually look at, again, is there a process with time from burning? The highest diversity in both cases is with our July sites. So that, again, is not what we were expecting with time since burn. And there's, again, this interplay between burn and control, not necessarily having the same consistent results. We also looked at whether or not community composition was different between our burned and control plots to, again, get at whether or not there are certain species driving any differences that we were seeing. And there was a little bit of a differentiation here. So we are seeing different species in our burn plots compared to our control plots, which could have impacts on when you're thinking about what functions certain species might be providing. I'm not going to walk you through the breakdowns there because it didn't end up playing out as much by what we would have expected by habitat associations. But I also want to mention that methods make a difference. And so our pitfall traps are really getting the ground foraging ant community. But if we think about what fire does, particularly in the system, it's burning away a lot of the leaf litter. And so a sampling strategy that's actually getting ants that are more in the leaf litter, they're not necessarily going down to the soil surface, might be better suited to actually looking at differences in our communities here. So we're expanding. We still have the little plots where the pitfall traps happen, but in a broader square around that, we have 4 corners where we're sampling the leaf litter and getting what ants are in there. And so this is for my second year. First and foremost, you can see that there is a difference in just, you see these different arrays of colors. That's just you see different combinations of ant genera in different, the soil community versus the leaf litter community. And that's no matter which treatment you have, that's just a sampling difference. We did have a little bit of a difference between treatments here, but you can already see that maybe the biggest difference is actually in our litter removal plots within these litter targeting Berlese funnels. And so just to look at these same results, the same types of results for the second year where we have the second method in it. We do see that where we have differences in our ant communities is not between the burn and control plots, but rather between either of those treatments and the litter removal when we're just looking at litter dwelling ants. And so that again might be that the burning didn't consume as much of the leaf litter as expected, or that there were other aspects about actually manually removing leaf litter versus not. And again, we don't see differentiation between our treatments in the soil dwelling community in the second year, but we do see differences with our litter dwelling ant community. But again, the biggest difference is coming from our litter removal plots, where we manually removed leaf litter. And community-specific compositional differences were harder to parse out as well. So there's a lot of overlap in which species are occurring in any treatment. And so that's a lot of results coming at you. But just to sum it up, I think some of the lessons that we've gained from this are that there's a lot of variety in ant response to treatment, but there was a lot of variation between year and location as well. And so far wasn't necessarily having a strong effect compared to these other aspects of our setup. But including abundance and weighting species by their abundance does impact your results. So when you're thinking about what diversity metrics you might be using, that might be something to consider for ants or for other groups as well. And importantly, choice of sampling method is very important for thinking about what results you actually are capturing. And so within this then, the question is, are ants a useful indicator in the system? Kind of, again, from one point of view, could be if you're seeing not a very strong effect of fire, maybe that's a good thing for management. You're not having drastic impacts on an important community. But also if we want to have some sort of group that is sensitive to effects, that's also something to consider. So I think that also brings in questions about just what are we thinking about when we're choosing indicator species, indicator groups, and what do we actually want to see versus where is that bringing in bias? So that's what I have for you today. I do want to just thank everyone who's involved in that work, but I also want to bring us back to and open up the floor for discussion. So if you have more questions for any of our speakers, I'd like to take a moment to just allow those to be voiced and responded to. So.
One question for you.
Yeah.
Your work was in control groups, which are far less potentially impactful than wildfires.
Yeah.
Has anyone done similar work comparing wildfire sites versus control sites?
So not yet, but there's actually another grad student who's at Cornell, and her work is mainly in California, but it is looking at effects of wildfires and particularly also indigenous fire management on ant communities. So keep a lookout.